Okay, so for those of you who are here, can you see the chat? Yes? Okay, cool. So I left, I'm gonna have an office hour on Thursday for 1 hour, so you guys can come pick up your exams. But you can also um, bring me your report if you want, and I'll check it out and let you know if I see any red flags so you can fix them before they're due, before it's due. But I'll make that announcement again once class starts in a couple minutes. If you guys have any questions right now, you're free to ask. That's a good question. No, so I'm gonna have one office hour on Thursday, and then I'm gonna have one virtual office hour, and it's gonna be, let me check here, Friday. It'll be Friday at 8 p.m. because I figure some students maybe are not done with their report yet. So I always try to make my office hours to help you guys as much as possible, and I think Friday night is a good day. Hopefully everyone is done by that time. And so yeah, I'm gonna have one office hour from 8 to 9 p.m. on Friday. You can bring your report and just share your screen with me, and I can point out if I see any like big red flags so that you can fix them before you submit it. So yeah, one office hour Thursday from 12:30 to 1:30 p.m. You guys can swing by and pick up your test, and then one office hour on Friday. And let's see. So here's the information for my office hour on Thursday. It's SNE 1335. Write it down if you want to make it from 12:30 to 1:30. You can bring your report, or if you just want to come and pick up your exam, you can. So I sent out an email, but a lot of my last class said they didn't get it. So hopefully you guys got it. Okay, and I think we're just gonna get started. So the first thing I'm gonna do is show you guys how I'm gonna be grading. We were instructed to grade like, pretty like sternly. So I wanna show you the best way to get an A. Research project sample. Okay, so basically I would start off like at this point, you guys are probably like almost done. So just go ahead and check it out. So like I would start off with this example because I'm gonna print out the example and I'm gonna have it right next to me when I'm grading. So like follow the exact format of the example. So like the exact organization, and like when something is bold, you should have it bold. This whole PBDID, try to get the website also. For the reference, try to have it in this format, then the website, and then, this. What I've seen so far, sometimes the website is down here. Things are just like not following this format. So try to make sure that your stuff really follows the format. Summary of the protein. Try to organize it the way that they did. Protein production and purification. So here, the red flags that I was seeing is that these things, like the information was there, but they were not in this order. Things were kind of just like everywhere. I don't want that. That's going to take me way longer to grade, and we don't want that. So what I asked students to do was just kind of like grab the chunk and move it around until the information matches and it's in order, because that way I can grade it quickly. All I want to do is read this, read yours, And if the information matches, I want to just give you all your points. Okay. And I want to do that quickly. So go ahead and make sure that all of your information matches. And then here for figure 1, this is the purified protein that was crystallized in the presence of the ligand. So here, notice that for every figure, you have your figure 1, and then you need a title. Every single figure needs a title, and that title should be in bold. So the whole figure 1 and title should be in bold, and then you write down where you took the figure from. And then over here, we have the 3D structure, method, and resolution. Those are important. And then you have A, and then you have B. So these should not be two different figures. Some students like use the whole page for this, but if you use PowerPoint, and I don't mean like the Google Docs thing, I mean like real PowerPoint, you can actually move things around. You can put the A, the B, and then you can copy and paste everything as a picture and bring it here. And then the figure will actually look like this. You don't have to do that. It's just if you happen to already know how to, that would be easier. And then one thing where I've been seeing red flags is that students don't know how to write this section. So make sure you pay attention to this. The way that it works is, again, you have your figure and then you have your title. So you should have the figure 2 and then title. All of that should be in bold. Then you have letter A, and that should be in bold. And so you need to describe to me, what am I seeing in A? So here it says structure as observed with a 3D view tool in the PBD site using the predict membrane tool. Protein and cartoon representative with amino acids colored according to their properties. Bound caffeine in stick representation. Gray discs represent the boundaries of the membrane. So do you see how we see like the stick representation that they're talking about? We see they're describing everything. And that's what you're supposed to be doing. And then we have B, and then you describe the same thing, okay? And then we have our FASTA sequence. So the way that they describe it here is figure 3, primary sequence of the protein, amino acids represented using the single code. Really, it's the single letter code. But yeah, like for example, all these H's down here, those are histidines. So let the reader know what we're looking at. But yeah, your sequence should follow this. A student asked in the previous class if there should be anything else here. What I would say is first follow this format exactly, and then go to the rubric and look at it step by step to see if you're missing anything. And then only if you're missing anything, then you can go ahead and add it to that section. And that's like the best way to get an A. Like you should get all of your points if you do this. And then over here we have the sequence. And then figure 4, sequence information shown in the PBD webpage. A more detailed description is in the next page. And then here we have the more detailed description. So here some students didn't know like what to write. This information should be in the paper. So remember, you were instructed to go to a website, type in the name of a protein, and then get all the information from that protein. But normally, every single protein is associated with a research paper. So those are the people who found the protein 3-dimensional structure. So that research paper should have all of the information that you need, like all of this information down here, if you go to the results or the discussion section. And then another student said that he went to office hours and the professor told him that you can basically describe anything that you're seeing here, like this secondary structure. That's something that we learned about in this class. So you can talk about that for this protein. Ramachandra and outlier, we learned about that. Hydropathy, how hydrophobic is this protein? So things like that, those are perfect for putting down here. Okay, we have something in the chat. It says, for the FASTA sequence, should we translate the letters? For example, how you pointed out histidine. Should we do that in a little description under the figure? I don't think you should do that. Like we already know what the letters are, like which amino acids they represent, and that would take you a long time. Like you shouldn't have to do that. Like I think everyone should be able to just find the sequence. We don't want you to spend hours doing this. Just grab it, copy it, paste it, and continue. But yeah, good question. Okay, so then the last figure is the ligands. So here's figure 5, caffeine, and its interaction with residues in the binding site of the adenosine receptor. So you notice how that's in bold. The title is describing what we're seeing, and then you describe this and where you got it. Okay, so I just wanted to give you a quick run through and advice on how to get an A. And then, yeah, if you bring it to me during office hours, either in person on Thursday or via Zoom Friday night, I'll be happy to look at it and just look to see if there's any red flags, since I know you guys are really stressed out about this. All right, so if anybody doesn't, if you have one more question, we can go ahead and ask it, but we're going to get started with our discussion for today. Okay, looks like we don't have any more questions, so we'll get started. So this discussion is really important. There's a lot to remember. We're going to be going through the 10 steps of glycolysis. You have to know which enzyme is associated with each step, what you're starting with, what you're ending with, what type of reaction it is, phosphorylation, dehydration, whatever. I just uploaded this right now, like 10 minutes ago, because I added a few more things. So, yeah, so just pay attention. If you don't have a whiteboard, I would recommend getting one just to like really review this material before the final. So, okay, question number 1. Describe different mechanisms that can be used for modulation of activity of enzymes. So when here we're talking about enzyme activity. And again, there's 10 different ways to modulate the activity of an enzyme. So previously I used the example of an enzyme that would make pizza. So if I decide that I want to have pizza all week, I'll use that as an example. So the activity of an enzyme can be changed by altering the number of total enzyme molecules available. So for example, if I want to make pizza all week, I'm going to say, okay, I'm going to increase the production of the enzyme that makes pizza. So I'm going to make like 100 of them. And you can do that in steps 1 through 6. So steps 1 through 6 will modify the amount of enzyme. So I can make a lot or I can make a little bit. Now, steps 7 through 10 will modulate the activity of existing molecules. So at this point, for steps 7 through 10, you can no longer modulate how much enzyme you have. You simply have whatever amount of enzymes you have, and you have to modify the enzyme's behavior in different ways. Okay? So remember, steps 1 through 6, you can modify the amount of enzyme. And steps 7 through 10, you can modify the activity, the behavior of that enzyme. And then they give you examples that were talked about in class. And if you remember from exam 2, examples that the professor talked about, they were in the exam. So you definitely want to check out these examples from your lecture notes and go ahead and annotate them and everything. Hopefully they will make more sense once I cover this. Okay. So we're going to go through all 10. Okay, so again, steps 1 through 6 will modify how much enzyme we have. So the first step is extracellular signals. So for example, that would be me telling my body, hey, I want to make pizza all week. So the extracellular signal would bind to the receptor, and then you would have a transcription factor, and that would go in and tell the nucleus, it would say, hey, I need you to produce a lot of this gene. I need you to express this gene, the gene for the pizza enzyme, right? So extracellular signal and receptor transcription factors. That is step one. Step two. Okay, sorry. I had a coughing attack. Okay. So step two is transcription of specific genes. So if I want the pizza enzyme to be transcribed, here you have the transcription factor, and it's starting the transcription. So you have the messenger RNA being produced for my pizza enzyme. And if I want a lot, I can say, okay, make like 100 of them. And then step three is messenger RNA degradation. So the messenger RNA for my pizza enzyme, let's say that my mom comes over out of nowhere and she says, you know what, you're not having pizza this week, you're eating salad. So all of a sudden my body's saying, okay, we have to degrade all of this messenger RNA for the pizza enzyme. We're no longer making the enzyme. And then step four is messenger RNA translation on ribosome. So my mom decides to leave because she had an emergency. So she leaves. And so now my pizza's back on, right? So I'm going to go ahead and make here's a messenger RNA, here's the ribosome, and it's producing my protein. So this protein is my enzyme. It's my pizza enzyme. Okay, and then so for step number 5, we have protein degradation, ubiquitin proteasome. So we have in our bodies a proteasome degradation system. So I like to think of this as it's kind of like when that little gun that has the stickers at the store where you just attach the little stickers with a price to each item. So this is kind of the same thing. We have that in our bodies, but the sticker is ubiquitin. And when you attach those stickers to a protein, the proteasome will go ahead and degrade that protein. So let's say that after one day of me being happy eating pizza, my mom shows up again and she says, okay, no more pizza. You're having salad. So all of a sudden my body will have to degrade all of the pizza enzymes that I already made. And then step 6, you can sequester these enzymes in subcellular organelles, the endoplasmic reticulum. So let's say I'm like, well, I don't know if my mom's gonna be here that long, so I'm gonna go ahead and just save these enzymes for later. So that way, in case she leaves in like a day or two, I can go ahead and just reuse them and I don't have to start over to make them. So remember steps 1 through 6. This is how you can modify the amount of enzyme that you have. You can have a lot of an enzyme or you can make a little bit of an enzyme. Steps 1 through 6. Now, steps 7 through 10 is when we already have the enzyme. There's different ways of modifying the enzyme activity. So for number 7, we have enzyme binds the substrate. So when the enzyme binds the substrate, a reaction happens. 8, enzyme binds the ligand at an allosteric effector. So the ligand is binding at a different site other than the active site, and it's causing something to happen. Number 9 is enzyme undergoes phosphorylation and dephosphorylation. So here you add a phosphate group or you remove a phosphate group, and that will have an effect on the behavior of the enzyme. And all of these, those are the examples that the professor's talking about. So definitely check these out. The GLUT2, ATP, PFK1, pyruvate, all that. And all of that is in your lecture notes. Okay, and then number 10 is enzyme combines with regulatory protein. So you can have a regulatory protein that allows the enzyme to perform its function, or you can have a regulatory protein that inhibits the enzyme's function. Okay, so I hope that you understand these 10 mechanisms for modifying enzyme activity a little bit more. Okay. Okay, so now we're going to start talking about the glycolysis reactions. So for these, I highly recommend that you create like a table in Excel or something. So it'd be, or it doesn't even have to be in Excel. It could just be, I think in your Word document, you can also create a table. So something that looks like this, and then you're going to have 10 reactions. So I would do like all 10 reactions. And then so like right here, I would start off with like maybe, I don't know, do reaction numbers. So this will be reaction 1. You're going to have 10 of them. And then I would say like, what are you starting with? So what molecule? So we're starting with glucose. And then what molecule do you have at the end of the reaction? And then I would say like the enzyme. So have more columns. I didn't draw enough. So I would say the enzyme and then type of reaction. So yeah, I would recommend, because you have to know all of this stuff for the final, and like you want to make sure there's a lot of material. So try to organize it. But I'm going to go ahead and cover as much as I can. And I already gave you a lot of notes, so hopefully that will help you fill in the table. Okay, so glucose. This is the molecule we're starting off with. So remember glucose, carbon 1, 2, 3, 4, 5, and 6. You have to know where the hydroxyls are. Remember that whether the hydroxyl is down or up, that will change the whole molecule. If you draw it wrong, or on the final, they might give you the molecule, but if you may have to identify it. So you have to remember like where the hydroxyls are at each carbon for glucose. Also, so we're starting off with glucose, and this we're finishing with glucose 6-phosphate. So this is reaction 1. Reaction 1, we're starting off with glucose, and we're ending with glucose 6-phosphate. So this is really easy. The number 6 just means that carbon 6 phosphate. So we have the normal glucose molecule, but we added a phosphate group to carbon number 6. Okay, so who did that? Well, the enzyme hexokinase did that. Hexokinase added a phosphate group to carbon number 6 on glucose. So we started off with glucose and we finished with glucose 6-phosphate. Now, where did we get that phosphate molecule? It used to be ATP and it turned into ADP. Okay, so we got that phosphate from ATP. So we're using energy here. And this is an irreversible reaction. Okay, so there are a few reactions that are irreversible, 3 of them, under physiological conditions, and you have to know which ones they are. And after we go through the 10 steps, I'm going to tell you why they're irreversible. So reaction number 1 is irreversible. It is a phosphorylation reaction because something is getting phosphorylated. Okay, so reaction number 2, we are starting off with glucose 6-phosphate, and we're ending with fructose 6-phosphate. So these are basically the same, except that in glucose 6-phosphate, remember that we have the aldehyde at the end, the carbonyl is at the end. Okay, so this is an aldehyde sugar. And then for fructose, the carbonyl was moved to carbon number 2. And whenever we have the carbonyl at carbon number 2, we know that this is no longer an aldehyde sugar. It is now a ketose sugar. So a question similar to this was on the exam. So make sure you know your terminology. Okay. So that's all that changed. So this went from glucose 6-phosphate to fructose 6-phosphate. And who did this? It was the enzyme phosphoglucose isomerase. So this was an isomerization reaction. Remember, reaction 1 was a phosphorylation reaction. You have to know which reactions these are. So for number 3, so reaction number 3, we start off with fructose 6-phosphate, and we end up with fructose 1,6-bisphosphate. So this means that there are now 2 phosphate molecules, one at carbon 1 and one at carbon 6. So let's take a look. We have carbons 1 through 6, and there's the phosphate molecule at carbon 1 and the phosphate molecule at carbon 6. So which enzyme did this? It was phosphofructokinase. Phosphofructokinase. And where did the phosphate come from? It came from ATP. So this was an energy requiring reaction, and this is an irreversible reaction. So far we had reaction 1 and reaction 3 that are irreversible. And this was a phosphorylation reaction. So remember, reaction 1 was phosphorylation, reaction 2 was isomerization, and reaction 3 is phosphorylation. Okay, so now reaction 4. We are starting with a 6-carbon sugar or molecule. This is fructose 1,6-biphosphate. And now what we're doing is we're cutting this molecule in half. We are cutting fructose 1,6-biphosphate in half. So we're going to end up with 2 3-carbon sugars, and each of them is going to have a phosphate group attached. So the enzyme who takes care of this is fructose biphosphate aldolase. Fructose biphosphate aldolase. So the first molecule that we have has carbon 1, 2, and 3, and this is dihydroxyacetone phosphate. And then over here, the second is a glyceraldehyde 3-phosphate, and this was carbons 4, 5, and 6. But now that it has become its own 3-carbon molecule, it's now carbon 1, 2, and 3. So remember, the number 3 phosphate, that's what tells us what carbon the phosphate is under. So some students previously drew like the phosphate on carbon number 1, and that would be incorrect. The phosphate has to be on carbon number 3 because the name of the molecule tells us that. Okay, so now the next step is that triose phosphate isomerase. This is reaction 5. We have an isomerase reaction, and this dihydroxyacetone phosphate, this molecule, gets turned into glyceraldehyde 3-phosphate. So by the end of step 5, we actually have 2 glyceraldehyde 3-phosphates. So let's check out the molecules a little closer. So for this dihydroxyacetone phosphate, the phosphate was at carbon 1. Carbon 2 had a carbonyl, and carbon 3 had an alcohol. So with glyceraldehyde 3-phosphate, we see that the carbonyl is carbon 1, but over here was carbon 2. So those guys got to swap. And then carbon number 2 has an alcohol, and carbon number 3 has the phosphate group. So that molecule gets rearranged with an isomerase enzyme, and it turns into glyceraldehyde 3-phosphate. So now, when we start step 6, we already have 2 glyceraldehyde 3-phosphate molecules. So remember, carbon number 1 is a carbonyl aldehyde, carbon number 2 is an alcohol, and carbon number 3 is a phosphate. So for reaction number 6, we have enzyme glyceraldehyde 3-phosphate dehydrogenase. So dehydrogenase, that means that a hydrogen is going to be removed from this molecule. This is going to be an oxidation reduction reaction because you're transferring a hydrogen. So I went ahead and circled in blue this hydrogen over here because that's the hydrogen that is being removed in step 6. So we have NAD plus inorganic phosphate. And so we take this hydrogen and we go ahead and give it to the NAD plus, and we turn it into NADH. Okay, so we took the hydrogen from our glyceraldehyde 3-phosphate and we gave it to NAD plus and we turned it into NADH. So because this is an oxidation reduction reaction, we want to keep track of who is oxidized and who is reduced. So this NAD plus is the oxidized form. And this NADH, remember the electrons are carried in these H's. So this NADH has more electrons, so it is reduced. So you gotta keep your terminology straight. Now, at the end of reaction 6, we're left with 1,3-bisphosphoglycerate. So we removed that hydrogen from glyceraldehyde 3-phosphate, and we went ahead and added a phosphate group. Okay, so now we have a phosphate at carbon number 1 and a phosphate at carbon number 3. That's why there's 1,3-bisphosphoglycerate. Okay, now we have reaction number 7. So reaction number 7, we start off with 1,3-bisphosphoglycerate, and we end up with 3-phosphoglycerate. So the enzyme is phosphoglycerate kinase. And notice that in 3-phosphoglycerate, the only difference is that we got rid of a phosphate. So the phosphate at carbon number 1. So what happened is that we started off with an ADP and we went ahead and gave it the phosphate. So now we produced ATP. So this is an energy production step. And this is a phosphorylation reaction. So reaction number 8, we start off with that 3-phosphoglycerate. So the glycerate portion is referring to the carbonyl with a oxygen with a negative charge. This portion right here, that's 8. So that's why this is 3-phosphoglycerate. So 3 on carbon number 3, we have the phosphate. 3-phosphoglycerate is turned into 2-phosphoglycerate. So this is the same molecule, but the carbon was moved, or the phosphate was moved from carbon number 3 to carbon number 2. So that's pretty easy. That's how we know that this is an isomerization reaction. Nothing was added, nothing was removed. The atoms were just rearranged. So the enzyme who did this was phosphoglycerate mutase. And I hope you're seeing a trend here that most of the enzymes are named after either the reactant or the product. So the product here was phosphoglycerate. The enzyme is phosphoglycerate mutase. Okay, and then we have reaction number 9, and you start off with 2-phosphoglycerate. So the phosphate at carbon number 2, and you end up with phosphoenol pyruvate, PEP. Phosphoenol pyruvate. So here the enzyme was enolase, and what it did was enolase removed a water molecule from 2-phosphoglycerate, and it turned it into phosphoenol pyruvate. So if you remove a water molecule, where did the hydrogens and the oxygen come from? So I went ahead and circled them for you. Enolase took this hydroxyl group at carbon number 3, and it took this hydrogen at carbon number 2, and that's how it got its water molecule to leave. And now this is what we're left with, phosphoenol pyruvate. And that was a dehydration reaction. And then for step number 10, we start off with phosphoenol pyruvate, and we end up with pyruvate. So the only thing that happened there was that the phosphate group that was attached to carbon number 2, it gets removed and it gets added to an ADP molecule. And so we end up with ATP for step number 10. So this is another ATP producing reaction. And the enzyme that takes care of this is pyruvate kinase. So it's named after the product. Our product is pyruvate. So a lot of these molecules, you should know how to draw them. So remember, pyruvate has this carboxylic acid over here, but with no hydrogen. So carbonyl with another oxygen. Carbon number 2 has a carbonyl, and carbon number 3 is just a methyl group, CH3. And that's reaction 10, and reaction 10 is irreversible. So reactions 1, 3, and 10 are irreversible. And the reason why they're irreversible is because they have a negative delta G under physiological conditions. So if that reaction has a negative delta G under physiological conditions, then it is energetic. Oh, it's not letting me write for some reason. Let's see here. Oh, I was just being really late. Okay. Oh, sorry. My computer does this sometimes, and then, okay. Let's erase this. Okay, so basically under physiological conditions, if you have a large negative delta G, then that reaction is favorable and it will be irreversible. Once it happens, you can't go back. So all the other reactions, they have positive delta G's, so under physiological conditions, so they can be reversed. So reactions 1, 3, 7, and 10 have a very negative delta G standard, which means that those are exergonic reactions. So exergonic reactions release heat, and they're favorable and essentially irreversible under standard conditions. However, only reactions 1, 3, and 10 are irreversible under physiological conditions. So only reactions 1, 3, and 10 are irreversible under physiological conditions. So the rest of the reactions have a positive delta G, meaning that the reactions wouldn't occur in the direction of the products. So we talked about this last week. In order for the cell to make a reaction happen when it has a positive delta G, remember, if a reaction is energetically unfavorable, it will have a positive delta G. The cell does two things in order to make that reaction happen. So number one is that the cell will couple that energetically unfavorable reaction to an energetically favorable reaction, like ATP hydrolysis. Okay? So step number one is that the cell will couple the energetically unfavorable to an energetically favorable reaction. And the second thing that a cell can do is that it can keep the concentration of products low. So we talked about the product to reactant ratio last week. So the reaction of products to reactants will be kept low. And those are the two things that a cell can do to make energetically unfavorable reactions happen. Okay, so here we're going over the exact same thing that we just went over, but this way it helps some students understand what's going on a little bit better. So they just drew the linear molecule for glucose 1 through 6. Those are carbons 1 through 6. Phosphorylation of glucose ensures that pathway intermediates remain in the cell. So you might have a question like, why is glucose phosphorylated? And the whole reason why is to keep it locked in the cell until it gets turned into pyruvate. So reaction 1, phosphorylation occurs on carbon number 6. So some students asked, why doesn't phosphorylation happen at carbon number 1? And the reason why is because there's a carbonyl group there and we need an alcohol. So where did the phosphate come from? It came from ATP. And so here we go, carbon 1 through 6, and we see that it's phosphorylated at carbon number 6. So this is glucose 6-phosphate. Now, reaction number 2 is an isomerization, and it moves the carbonyl to carbon number 2. So here we have our carbon number 1, which was an alcohol, or before it was a carbonyl. But we just moved that carbonyl to carbon number 2. And so now we have carbon number 1, we have an alcohol. So remember, the reason that we want an alcohol here is because we want to be able to add a phosphate, because later we're going to break down this molecule in half, and each one has to have its own phosphate group. So this molecule is called fructose 6-phosphate. And yeah. And then for reaction number 3, we finally add the phosphate group to carbon number 1, and we take it from ATP. That's why we have ATP turning into ADP. So now that we have our phosphate at carbon number 1 and carbon number 6, this molecule is called fructose 1,6-bisphosphate, and we can go ahead and cut it in half. So it says the carbonyl at carbon number 2 facilitates the carbon-carbon bond cleavage at the right location to yield 2 3-carbon products by the reverse of an aldol condensation. So a lot of students didn't know what an aldol condensation was. So a condensation reaction is when a water molecule leaves and a bond is formed. So this is the opposite of that, because we break this bond. And we're left with 2 3-carbon molecules. We have dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So then here we have those 2 3-carbon molecules, but we need both of them to be glyceraldehyde 3-phosphate. So we actually turn dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. And then for step 6, that's the oxidative phosphorylation step, where we have NAD plus coming in with phosphate and NADH. And so that is where carbon number 1 gets a phosphate. So now this molecule is 1,3-bisphosphoglycerate. And we took the hydrogen that used to be here at carbon number 1, and we gave it to NAD plus to make NADH. Remember, NAD plus is the oxidated form, and NADH is the reduced form of that molecule. And then for step number 7, we have ATP production. So remember, this is where we take the phosphate that we just added to carbon number 1, and we give it to ADP to produce ATP. And we're left with 3-phosphoglycerate. And then for reaction number 8, the remaining phosphoryl group moves from carbon number 3 to carbon number 2. So in the book, there's a typo, and I believe the typo is also there in the lecture slides, but you can clearly see that this phosphate was at carbon number 3, see carbon number 1, 2, 3, that's where the phosphate was, and it gets moved to carbon number 2. So they just kind of had them backwards. And so we're left with 2-phosphoglycerate. And then for step number 9, it says dehydration activates the phosphoryl for transfer of ADP. And this is where that water molecule left. And then we end up with phosphonyl pyruvate. And then for step number 10, we had ATP production. So that's when we took that phosphate group from carbon number 2, and we gave it to ADP to make ATP. And that left us with pyruvate. So remember to make your table and organize everything. What are the energy producing states? What are the enzymes that take care of each reaction? What type of reaction do you have? Is it a phosphorylation? Is it an isomerization? Is it an oxidation reduction? Is it reversible? Is it irreversible? Okay. So hopefully all of that is a little bit easier now that we went over every single step. This, I'm going to let you go through it. I guess I can walk you through this. We have 7 minutes left. So if you have any questions, right now is the time. I'm just going to get started on going over this, but you can interrupt me if you like. So describe the catalytic mechanism of the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. In this reaction, which amino acid is a nucleophile? Who is oxidized and who is reduced? Who is a high energy compound and why is it a high energy compound? Okay. So we start off, this is glyceraldehyde 3-phosphate dehydrogenase. So this is an enzyme and this is the active site. So notice that there's NAD plus hanging out right there. And then over here, we've got cysteine. So normally there's an SH, but the hydrogen is gone. So that's why we have S minus. So formation of enzyme substrate complex. The active site cysteine has a reduced pKa 5.5 instead of 8. So remember, the pKa value is the pH at which you lose or donate your hydrogen. So normally cysteine would donate its hydrogen at a pH of 8. But here in this location, it donates its hydrogen at a pH of 5.5. And remember that our physiological pH is about 7. So that is why it already donated its hydrogen and it has a negative charge. So you have your substrate that comes in, and that's glyceraldehyde 3-phosphate. So here it is. We have carbon number 1, which if you remember, it is the aldehyde. So there's a carbonyl with a hydrogen. Carbon number 2 has an alcohol, and carbon number 3 had the phosphate. This is glyceraldehyde 3-phosphate. So it's going to attach from carbon number 1 to the cysteine. So that is a covalent thiohemiacetal linkage forms between the substrate and the sulfur group from the cysteine residue. So this is a thiohemiacetal linkage, and it's just this sulfur group attaches to that carbon. So step number 3, the enzyme substrate intermediate is oxidized by the NAD plus bound to the active site. So when something is oxidized, it means that you're removing electrons. So if you notice here, this NAD plus is stealing this hydrogen, and along with that hydrogen, it's taking the electrons. And that is why this enzyme substrate intermediate is being oxidized, while the NAD plus is being reduced to NADH. So you see that it has been oxidized, so it created this carbonyl, and now you have the NADH. And for step number 4, the NADH product leaves. So that's the first product, NADH. Now it's happy that it has become reduced, so it leaves. And it leaves the active site, and it is replaced by another NAD+. So it's replaced by another NAD+. And then here we have a phosphate, inorganic phosphate coming in. We have an NAD+ coming in, and we have NADH leaving. And so the covalent thioester linkage between the substrate and the enzyme undergoes phosphorylation. That's attacked by phosphate, releasing the second product, 1,3-bisphosphoglycerate. So that inorganic phosphate came in and it attacked at that carbonyl. It attacks that carbon. So now this is carbon number 1 is going to have a phosphate group. Carbon number 2 is going to have a hydroxyl. And carbon number 3 still has the phosphate group. So this 3-carbon molecule now has 2 phosphate groups. And when this phosphate attacks this carbonyl, then it lets go. So it's going to let go from the enzyme binding site. And so that will be the second product. So I don't know what questions you'll get on the exam, but if they ask you anything about what is the first product, that's NADH. NADH is the first product. It gets its hydrogen, it gets reduced, and it leaves. And then the phosphate will come and attack, and that will cause it to release the second product. And when it has both phosphates, it's called 1,3-bisphosphoglycerate. So that's the second product. And then that's it. That's what glyceraldehyde 3-phosphate dehydrogenase does. Okay, and it's saying cysteine behaves as the nucleophile. So cysteine right here is the nucleophile, and G3P is oxidized to 1,3-BPG. NAD plus is reduced. So yeah, whoever is losing the hydrogen becomes oxidized. So that was glyceraldehyde 3-phosphate, that's G3P. And then NAD plus was reduced because it took the hydrogen. Energy released by carbon oxidation is converted to a high energy compound, 1,3-BPG. So this 1,3-bisphosphoglycerate, that's a high energy compound. The standard free energy change of hydrolysis is minus 49.3 kilojoules per mole. Its hydrolysis is very favorable. It has a very negative delta G because the products 3-bisphosphoglycerate and phosphate can adopt resonance structures that helps stabilize the negative charges. So the question asked us why. First of all, who is the high energy molecule that is produced? It's 1,3-BPG or 1,3-bisphosphoglycerate. Now, why does it have high energy? And it's because it is resonance stabilized. The fact that it is resonance stabilized gives it a really high negative delta G of 49.3 kilojoules per mole. So you have to know that. And then this is how that happens. And then that's it. That's all the time we had for today. Does anybody have any last minute questions? If you don't, please make sure that you review the rest of this worksheet. It is really important.